Hi everybody, welcome to part 2 of our look at the nervous system. In this particular one we're going to focus on how a signal travels down the axon of a neuron. So let's jump right in. First and foremost, we're going to talk about the resting membrane potential that exists in a neuron. And what this means is sort of the normal levels and concentrations and the normal voltage that sits before a signal comes in to actually fire a neuron, meaning a signal that gets sent down the axon to elicit a response. And when we talk about potential, just like when we talked about potential at any time over the course of the year, in this case we're looking at electric potential. We're talking about differences in the charges across a membrane. And these differences in charges result from ion concentrations, and we get this electric charge, which is kind of cool. So the cytoplasm that's near the membrane has a slightly negative charge with respect to the fluid that's outside. And in the case of our neuron, okay, it is highly concentrated with sodium ions outside and highly concentrated, way more, with potassium inside. So the sodium is way higher outside than in, potassium way higher inside than out. And this is going to play a role in how that signal gets sent down. The voltage actual reading on it is around about negative 70 millivolts in terms of the potential at any time across that membrane. And this is where it sort of sits at rest. And there's energy in that difference, okay, because of the movement of molecules. There is some energy in that difference in voltage that's going to allow for, let's say, enough of a signal to be sent to actually trigger at the trigger zone. So graded or local signals, meaning ones that come in or local, can decrease that potential, okay, and actually lower the potential. And as it lowers, meaning going up, it could reach a threshold which will then fire a response. So let's talk a little bit about what's going on inside and outside. So as I mentioned, high amounts of potassium inside, high amounts of sodium outside. And through ungated channels, these are protein channels that have nothing barring them, there can be a little leakage, if you will. So little sodium coming in, little potassium floating out just due to natural diffusion, which can occur. Okay, so let me showcase a little bit of this. Here's what we've got going on. So we've got these ungated channels that allow for just regular old diffusion to happen, okay? So right here, passive transporters, ions can just literally leak. So if I've got potassium here, potassium can just head on out because there's a higher concentration inside than out. Same thing goes for sodium. Sodium can move in because sodium is much higher outside than it is in. Now the sodium-potassium pump is going to play a big role. We've heard about this before because we've talked about its role in active transport. Well, here's where we're really going to get to know and love the sodium-potassium pump. So this pump is important because it will restore that resting potential. So as soon as we get movement of these molecules to the point of where we get too much potassium going out, too much sodium coming in, this pump here will actually reestablish that very distinctive concentration gradient that's going on here, okay? And that's what will happen. Okay, so this is actually a lovely protein descriptor of the sodium-potassium pump, so we can see a little bit of what it looks like on a molecular level. Okay, so again, remembering these are proteins. Proteins are coded by genes, right? So a gene coded for the shape of this protein that sits inside the membrane of our neurons. So here's the idea, right? So cell interior, cell exterior, okay, that's the outside. Okay, what I have, low sodium, outside high sodium, okay, outside high sodium, inside the potassium sodium. The potassium sodium pump is actually pushing against the gradient, okay, so it's actually going to move, okay, from low to high versus from high to low, and it moves 3 sodiums for every 2. That's the idea of the resting potential. Let's get a look at what actually happens when a signal moves down a neuron, which we call the action potential. Okay, really important, okay? We can actually use electrodes on a neuron, okay, to actually stimulate a response, and they did this in fish. Again, remembering we are in chapter 34, okay, so you can always go and use this as an additional resource, okay? you can actually measure the fact that you have, even though both potassium and sodium are positively charged, because of the relative difference in the amounts that there are, like there's way more sodium outside concentration-wise, so there's way more pluses outside than there is pluses inside. So we kind of refer to that as being it's way more positively charged outside the cell than it is inside. So it's not that it's negative inside, it's just less positive, I guess you could put it. So signals stimulate at the input zone, and if the signal's not strong enough, no signal will continue. And that happens here. Here's my negative 70 millivolts that I mentioned before at resting potential. So cells, there's really no signal, do. Well, let's say that there's enough of a graded signal, enough of a shift in these molecules where it starts to rise. Okay, fine. It's actually, starting to lessen that voltage, okay? If it hangs out somewhere within this area, there's going to be no signal. You need enough of a signal to actually elicit the response, and it needs to pass what we call the threshold. And if it surpasses the threshold, boom, done. It is an all or nothing response, and the signal gets sent, and there's no turning back once it fires. So we get this really, really important that it has to pass threshold for there to be a signal to propagate along. So the signal, okay, will cause gated channels to open, and sodium will flow in. Okay, so if I've got my little membrane here, right, and I have a bunch of sodiums out, okay, first thing that happens is the gated channels open and sodium comes in. Obviously, this is going to start to shift, okay, the potential, because if I've got all these pluses going inside, okay, I'm decreasing that concentration, because right in here I've got all kinds of potassium in here. Okay, so there you have it. Okay, here's all of those pluses. These are just representative of a region of more positive than inside the cell. So when the signal's going up, gated channels are opening, so boom, in comes sodium. Okay, if we have a stronger, very lasting stimulus, so we get a ton of channels opening, okay, get them to open. So let's think about what's going to happen. Okay, the threshold is surpassed or reached, right? Okay. The electric potential is going to move further away from the resting potential, further. We hit threshold, bam, we're going to get a signal. So here's sort of the way the events goes. We reach threshold at the trigger zone, electric potential is reached, and we set levels, okay, from the resting potential. So that's kind of the route things have to take, okay? We cause a shift, and then we have to reset it. So here's the way it looks. As soon as that threshold's reached, every single gated channel opens in that area, and sodium rushes into the neuron, okay? That's what we have here. And as a result, I have now shifted my charge, where it's now a more negatively charged area here, more positive inside, because all of those sodium is coming in. Okay? That causes this reversal of polarity. Okay? And think of how long it's telling me, it's taking me to explain this. This event happens in the duration of a millisecond. Okay? It takes for this to happen. Okay? So all of these channels open. As soon as that happens, okay, this idea is positive feedback. I am pushing something away from rest, away from the norm, to elicit a response. And eventually I'm going to have to reset it. Okay, that's the idea of that positive feedback. When this happens, this sets off a chain of events that is doing the same thing all the way down this membrane. As these ones open and we shift, then this one's going to open, and then this one's going to open, and then this one's going to open. And it sort of happens in this very chunked, succinct manner, so that we get this shifts, and now we're positive down here and negative here. Positive, negative, and it works its way down. Okay, and it's all or nothing. You have the action potential or you don't. And so here is the idea. Okay, here, this is all gated channels open. Okay, Na plus in during this point, okay? When we reach this, this is the neuron, which I'm about to talk about, trying to reestablish. This is going to send potassium out, okay, at that result. So let me talk about that for a second. The restoration of the resting potential. Okay, I've opened up all these channels. Now I have all of this sodium inside the cell, okay, way off of what the resting potential is. Notice I'm way up here, okay? -70, that's the resting potential. So I'm way up here. I've got to reset it back down. I've got to get it back down to here, okay? How do I do that? Okay, well, the immediate answer, sort of the quick fix answer, is for potassium to flood out through its gated channels, okay, through diffusion. I've got all of this potassium in here. Let's make it rush out. Okay, because if I do that, I'm resetting and reestablishing my positive here. Notice because here I'm negative, and that's going to keep going down. So as this goes down, as sodium rushes in, oh, I'm going to take potassium and I'm going to move it out. And that just restores temporarily. Here, I'm restoring my resting potential. Okay, but I can't have a signal without having sodium rush in. And if all the sodium's inside my cell, okay, I've got a problem, okay? So not only do I have to reset the resting potential, I have to reestablish the concentrations where I have all my sodium outside and my potassium inside. Here's where we get the sodium-potassium pump. It is going to take and actively transport all of my sodium that's outside, pull it in, or excuse me, all my potassium outside, pull it in, take all my sodium that's inside, and pump it out. But of course, this is active transport. I'm moving against a gradient. So think about that. What does that mean for the cell? What does it have to use to actively transport substances? I can hear you. Everybody say it with me. It needs ATP. Good job. Okay? And that is why sometimes you say it takes energy to think. It takes energy. It literally takes energy for us to think. To send a signal requires energy. So by this point, okay, here, all right, I have reestablished, okay, my charge, reestablished the potential here. But what I haven't done yet is reestablished the concentrations. And to do that with the active transport, we have what we call the refractory period. This is a moment in time where, as we're resetting those concentrations, sodium back out, potassium back in, okay, there can be no signal sent down the neuron, okay? This allows for signaling down a neuron to remain a one-way street, because what you don't want to have is the signal that's going this way get bounced by one trying to go the other way, okay? Nothing would happen. You'd go nowhere fast. So you've got the signal coming down, this one here, okay, that wouldn't work. Nothing would get accomplished. So because of the refractory period, it makes a moment, okay, where it's impossible for that neuron to receive a signal at that little place on it, okay? And that allows it to maintain its movement in a single direction down the neuron. Kind of cool, okay? What's really neat about these is that they're self-propagating, meaning once one goes, it's a chain reaction, and they all go, and they'll all go in succession from one another. Okay, and this happens by chunks down an axon. Okay, axons are not all wide open. Okay, this span of sodium, right? Okay, out to the adjacent membrane area causes gated channels down in the next junction to open up. And then those cause the ones down to open up. So it's this, you know, again, movement down in a one-way street, okay? Because they're self-propagating and because they happen in these chunks, there is no strength loss in the actual signal. And action potential has a very distinctive no strength lost, okay? The reason why we can do things like, you know, pick up heavy things versus not, comes from how many of these we fire at a time, not a single one, okay? And this comes from the idea of irritability of the membrane. It's very sensitive to these changes and can make these things happen as they go down. Okay, so again, our reminders of how the sodium potassium pump works. Okay, if I'm inside the cell, okay, we get this shift that occurs. Sodium potassium pump is going to move things against that concentration gradient, again, to restore the concentrations. Okay, so from there, you know, that's pretty much it. I just really want to spend a little bit of time, just real quick, and kind of showcase. If I have a neuron, right? Okay, so here's, you know, kind of your standard neuron, right? Okay, so I got my dendrites, and then here's my axon. Now this is where, and then axon, we'll call these, you know, we got a little branching axon endings. Okay, my axon here is insulated with these myelin sheaths that run down the lengths. They're literally like insulators. Cells called Schwann cells secrete this myelin, okay? And it's called a myelin sheath, okay? So you have this myelin sheath, and when we're talking about here, so the trigger zone gets hit with its signal. So coming into the dendrites, here's my direction, okay, of my signal. And here, I'm going to have an action potential. All of these gates are happening. Everything happens there. This shift is going to cause this area, what we call the node, to have an action potential. And all I'm doing is drawing that little graph. Remember the graph here where we had the resting potential. It spikes up and then there. Okay? And there's my threshold, let's say, right there. Okay? So this has its own action potential in here, which will then propagate the signal down to the next node, having its own action. So which then propagates it down to the next node, propagating it down to the next node, propagating it down here to the next node, and so on, so on, so on, until we get to the very end. And what this results in, okay, so what does all this mean? All this results in releasing material out into a synapse. And all a synapse is, if this is the ending of my axon, I get a dendrite of the next neuron, let's say, right here. This is an important space, and that's called the synapse, and we're going to get into that later on. So what I want you to see, there's my nucleus of my cell body. So here, signal comes in this direction, trigger zone hit, all these little action potentials all the way down. And what's neat is that these myelin sheaths actually speed up a signal being sent. They've done studies where you have an unmyelinated axon. The signal is actually slower down an unmyelinated axon than it is in a myelinated one. And I think it has to do with the pooling. You know, there's a pooling of sodium that can happen in this area versus having it all spread out. So in that sense, that's how we get a signal down. We'll spend a little fun time with it as we come back and work with this a little bit more. So I know it's a lot. We'll have a good Wisco, you know, I'm sure we're going to have a good Wisdisco on this and really talk it through. So have a good night. Go back, refer to anything if you need to. Use your book too, and we'll catch you later in class. Take it easy.